So yeah, welcome to this lecture. Before we get started, I would like to briefly introduce myself. I'm a PhD student at Safari Research Group, and I work with Onur in this group, and here is directions that I'm interested in. So of course, a computer architecture, specifically in large-scale bioinformatics applications, and storage systems and their interactions, and overall other modes of near data processing and emerging technologies. And this is my contact information. So in this talk, we will particularly focus on specializing the storage system for genomics and metagenomics. So we've been working in this direction for a while, and the MegIS work that I will talk about in detail in this talk is an example of one of the designs we've made in this direction. So to motivate this direction a little bit before diving into MegIS's design. So as you have seen in this lecture several times probably, there's a lot of importance for genomics and metagenomics in many different critical use cases in medicine and understanding microbial environments and disease outbreaks and evolution, etc. And in the end, so we want to analyze this DNA for different organisms, for different purposes. And this is whole different strings of the different organisms' DNA. Let's say we extract them, and based on similarities and differences these strings have compared to each other, we would be able to make biological or medical conclusions. And these are the devices with which we are generating these sequences, and the throughput of them are improving over time. And they generate this DNA data with really large throughput. But the problem that we have overall in genomic and metagenomic analysis today is that the rate at which we can generate this data is much faster than the rate we can analyze it in conventional systems. So that is very important to rethink what type of systems we want to design to efficiently analyze this data. So this is a very simplistic view of systems today. So storage system, memory caches, and computation units. Due to the importance of this field, these metagenomics and genomic fields, there's been a lot of effort into accelerating them. So all this data needs to come from storage system to memory to caches and computation units. So therefore, it incurs computation overhead and also data movement overhead from storage system. And as we discussed, a lot of effort has been to accelerate these things by developing efficient heuristics, efficient filters to filter the data that does not need to be analyzed using expensive computation or using different accelerators. And also a lot of attention has been put into it from industry. And of course, these solutions provide a lot of benefits, and a computation overhead has been significantly alleviated. Of course, there's still more room to alleviate it, but there's been a lot of advancements made here. But what we show here in our research is that there's the data movement overhead from storage systems still persists, and its impact actually on end-to-end performance can become even larger when other overheads in other parts of the system get alleviated. Right? So that is the motivated by this point. Our goal in this line of research has been to improve performance of metagenomic and genomic analysis by reducing large data movement overhead from the storage system. And through our designs, we want to ensure that we design cost-effective systems and maintain the high accuracy of these applications, because this is very important for many critical use cases. So this is the overall goal that we have in this research direction. But in this lecture, we are going to focus on one of these designs, which is called MegIS. So we have recently published and presented this work in ISCA 2024. It is MegIS is high performance, energy efficient, and low cost metagenomic analysis with in storage processing. So this is the outline of my talk. I will give a brief background on what metagenomic is, and then talk about motivation and goal and the design and evaluations. But so what I would like to say is that please feel free to interrupt me if you have questions, because some content might be new in terms of required backgrounds. I try to provide it, but if there is some part that needs further clarification in any part, feel free to interrupt and ask questions. So metagenomic is the study of genome sequences of diverse organisms within a shared environment, for example, in blood or ocean or soil, etc. And it overcomes the limitation of traditional genomics in a sense that it bypasses the need for analyzing each of these individual species in isolation which might not be possible in several critical use cases. Let's say if we have a complex sample that in which organisms are already mixed in it, it would not be possible to go in a lab, and it would not always be possible to go in a lab and analyze them individually. So yeah, by overcoming this limitation, metagenomics has led to groundbreaking advances in many fields, such as precision medicine and understanding microbial diversity of an environment, and discovering early warning signs of communicable diseases, right? So since the species that are in a metagenomic sample are not known in advance, metagenomic analysis typically requires performing several tests to get an understanding of what is in the sample. So now I'm showing the DNA of these organisms here, for example. So the first step is to do the preparation of input queries. So we have here, we extract some k-mers, these are sequences of length k out of these DNA sequences of organisms. And the next step is presence absence identification, in which we want to see what is in the sample. So we look up these k-mers into a large database containing information of many species. And this database can actually be very large, so more than 100 terabytes in emerging databases. Then based on that, we would understand, okay, these are specific species that are present in the sample. And there's a third optional step of abundance estimation, which some applications require to find out the relative frequencies of occurrences of these organisms in the sample. Okay, so since metagenomic analysis typically requires accessing these large amounts of data, we perform a motivational analysis in this work to understand the impact of storage system on performance. So we perform a case study of performance of metagenomic analysis tools with various state-of-the-art SSD configurations. And compared to this idealized scenario in which no IO overhead exists, we show that compared to this, in both performance optimized and cost optimized SSDs, we see that IO data movement overhead causes significant performance overhead. So there is this large headroom here. And we also further analyze in our work that IO becomes an even larger overhead in systems where other bottlenecks, such as computation and main memory, get alleviated, right? So this IO overhead, due to accessing this large amounts of low reuse data, is really hard to avoid. So one might think that it is possible to do some sampling techniques to shrink the database sizes. And of course, there's been some works in this direction to make it more manageable to analyze these large databases. But these works reduce accuracy to some levels that would be unacceptable to many accuracy critical use cases. And another potential solution might look like to keep all data required by metagenomic analysis completely and always resident in main memory. So such a solution is energy inefficient, costly, and unscalable and unsustainable due to two reasons. First is that database sizes that are already large, as we discussed, increase very rapidly, doubling every few months. And the other reason is that regardless of individual sizes of these databases, different analysis might need to access different databases. So it will be very unscalable and unsustainable to perpetually keep all of them, all possible databases that need to be accessed in memory at all times. So motivated by these points, as we discussed, so I reiterate the goal again, which applies specifically also to this work as well. So it is to improve metagenomic analysis performance by reducing data movement overhead from the storage system in a cost-effective manner and with high accuracy. So we do not want to make the compromises that I mentioned in the previous slide, right? So while in storage processing can be a promising direction to achieve this goal by directly analyzing data inside the storage system, it is very challenging to actually use it here because no metagenomic analysis tools can run inside the storage due to limitations of the SSD's modern hardware. So none of these existing tools can run effectively there because of these limitations. Let's see. So latency of NAND flash chips is very large, and then the DRAM capacity that we have inside the modern SSD is typically very small, and its bandwidth is also limited. So by addressing these challenges in our work, we propose MegIS. So MegIS is the first in storage processing system for end-to-end metagenomic analysis. And a key idea of MegIS is to do cooperative in storage processing for metagenomic analysis, which is based on hardware-software co-design between the storage system and the host system. So we want to efficiently leverage the best of both worlds in a sense to be able to perform end-to-end metagenomic analysis. So let's revise these steps and then see MegIS' steps here. So it's in here, MegIS step 1 would be preparation of queries. Step 2 would be presence absence identification. And step 3 would be abundance estimation, right? So to leverage and orchestrate the capabilities of both of these systems, MegIS includes hardware-software co-design in 5 general directions. The first one is task partitioning and mapping, such that we ensure each step of metagenomic analysis executes in the most suitable system. So as we will see later, step 1 will run in host system, and step 2 and 3 will run in the storage system. We will discuss in more detail later why. But the second direction of hardware-software co-design we do is data and computation flow coordination, such that while we are leveraging the best of both worlds, we would also ensure that we minimize the overhead of communication between these two systems, and also ensure that this communication does not lead to excessive writes to the NAND flash chips that would hurt the lifetime of SSDs, right? And the third direction is to have storage-aware algorithms. Because of the very different hardware context we have there, the way we need to think of optimizing algorithms there is fundamentally different from the way we would optimize it in the host system with its very different capabilities, right? So then we have done storage of algorithmic optimizations to enable efficient access patterns to the SSD. And the fourth direction is to have lightweight in storage accelerators so that we minimize SRAM and DRAM buffers and the spaces that are needed inside the SSD, because as I discussed, there's hardware constraints in there. And finally, we have data mapping scheme and flash translation layer FTL, which is specialized to the characteristics of metagenomic analysis. And that also, it helps us to enable SSD's full internal bandwidth. So these are these general directions. Let's see how they are used in some of the steps of MegIS. So let's get started with step 1. So MegIS works with sorted data structures so that it would avoid expensive random accesses to the SSD. So it means that the databases and all these query k-mers need to be sorted. So therefore, the first is the preparation of input queries requires extracting k-mers from the sample, metagenomic sample, and sort these k-mers. Database k-mers are made offline and are sorted offline. So now, MegIS executes this step in the host system because it benefits from the larger DRAM capacity that typically we have available in the host system and more powerful computation. And it incurs because of that, it would also incur fewer writes to the NAND flash chips than compared to the case that we were to execute this step in the SSD. And by effectively leveraging the host system for performing this step, we would be able to overlap this step with the step 2, which, as we will discuss, will be executed in the storage system. So to execute this step efficiently in the host system, MegIS needs to ensure some things. First is to avoid significant overhead due to data transfer time between different steps, and also to minimize performance and lifetime overheads, even when the host system cannot hold all the query k-mers that we extract from the input sample. So now let's look at the design of this step a little bit. So we read the input queries, we extract the k-mers. So MegIS divides k-mers into independent partitions based on their alphabetic order, right? So this way, we can overlap operations that we do on these independent and like disjoint partitions, such that when we are performing sorting in one of the partitions in the host system, we can transfer and perform step 2 operations in the previous partition. So this way, this communication time between them gets hidden really nicely. And given that the database is sorted already, and this, let's say, alphabetically disjoint partition is going to the storage system, it can already start working with this without needing to wait for all other partitions, right? So there are, of course, a lot more optimizations about how we handle the case that the host DRAM is cannot hold all the partitions at once, and I invite you to check the paper to learn more about it. But now also let me summarize the details of step 2 a little bit more. So in step 2, we have presence, absence identification. So we have the k-mers. We want to now see what species these things belong to, right? So we first need to identify the common k-mers, meaning k-mers that are both in the query k-mers and also in the database k-mers, right? And after that, we want to retrieve the species IDs of these common k-mers, meaning figuring out what exact species these k-mers are coming from. So we execute this step in the storage system, in the SSD, because it requires accessing large amounts of data below reuse, that really large database that we talked about. And it also involves only performing lightweight computation. So it's not that it does not require expensive resources, hardware resources. So to execute step 2 efficiently in the SSD, MegIS needs to leverage internal bandwidth of the SSD efficiently, and also not require expensive hardware inside the SSD, meaning not rely on large DRAM bandwidth or DRAM capacity or large cost and costly logic units. So to identify the common k-mers, as we discussed, we need to see what is in the database and what is in the query k-mers. So relying only on the small DRAM with its limited bandwidth in the SSD to buffer the k-mers that come from the database and the k-mers that are coming from the sample query k-mers that are coming from the sample analyzed the host system, and then reading them out to perform analysis on them and find their intersection, and finally writing them, the common k-mers in the DRAM. It can limit it can pressure this limited internal DRAM bandwidth of the system. So what we do is actually to compute directly on NAND flash data streams. So we employ a technique that enables us to do that. Okay, so that is good, and we don't need to rely on the DRAM inside the SSD. But the issue is that we would then need to have large buffers for each of these channels. So by leveraging key features of this step in the metagenomic analysis and how the exact access pattern work, we managed to design the hardware such that we can reduce the buffer sizes based on these application features. So we can only so we only need 2 small k-mer registers per each channel. So that actually enables going over that really large database to find all the k-mer intersections with minimum hardware cost. So exactly what the features of the applications are. Also, again, I invited to check the paper to see that why only these 2 registers would enable us to buffer enough data so that we can access the data efficiently for these intersection units. So, okay, now we find the intersection between the query k-mers and these database k-mers and write the common k-mers in here. So now the next step is to retrieve the species IDs of these common k-mers by looking up a sketch database. So what is a sketch database? Has the list of these representative k-mers or sketches of different species and the species IDs of these k-mers. So by looking up these common k-mers in here, we would understand, okay, this is from this particular species whatsoever. So naively, storing these tables like this would be very space inefficient. And that is why there's been a lot of great effort put into representing these sketches in more space efficient manner. But the issue is that typically accessing these data structures require a lot of pointer tracing operations, which would be very slow inside the storage system due to the long latency of NAND flash memory. So what we come up with in this work is to have an intermediate between these. So we call it k-mer sketch streaming. So it is significantly smaller than, let's say, conventional way of storing them. still larger than the state-of-the-art approaches to store these sketches, but we find that that is a suitable trade-off to make for the storage processing paradigm because it is enabling us to perform these sketch lookups only using streaming access patterns. And that is what, again, I would like to emphasize the point I mentioned earlier, that sometimes we need to kind of fundamentally rethink how we optimize algorithms and what, let's say, optimization criteria we have to get the best of the underlying hardware. So yeah, I again invite you to check the paper for more details about this data structure and the algorithm. Okay, similarly, again, for step 3, please check the paper. We have more details about it, and also about the flash translation layer of MegIS and how data mapping actually works. So now let's go to the evaluation part. So we perform analysis of performance, energy, and power of the system based on the power and energy, the performance of hardware components and software components involved in MegIS. And we have these based on comparison plots. So the first one is performance optimized software. The second point is accuracy optimized software, which achieves better accuracy compared to the first one, but at low performance, and it has a much larger and more comprehensive database. And the last point is a PIM hardware accelerated tool that uses processing in memory to further accelerate the performance optimized baseline. And we analyzed MegIS on different SSD configurations, the cost optimized SSD and performance optimized SSD, which achieves higher bandwidth compared to the first one. And we analyzed different metagenomic analysis tasks. So I'm going to particularly talk about finding species that are in the sample. For that optional abundance estimation task that we talked about, you can check the results in the paper if you're interested. So for input sets, we have metagenomic samples with varying degrees of genetic diversity, low, medium, and high. Now let's look at the results. So we show that on a cost-optimized SSD, across different input sets, MegIS leads to, compared to both performance optimized and accuracy optimized baselines, MegIS leads to large speed up. And repeating this experiment on a performance optimized SSD, we also observe large speed up. So MegIS improves performance of both cost optimized and performance optimized SSDs. And here we see results compared to the PIM hardware. So similarly, we see that large speed up is achieved in both in systems with both cost optimized and performance optimized SSDs. Now I'm now showing the average energy reduction compared to different input sets and different storage systems. So let's take a look here. MegIS provides significant energy reduction over the performance optimized, accuracy optimized, and PIM baselines. And what I would like to emphasize here is that it achieves the same accuracy as the accuracy optimized baseline. And it is significantly higher than the accuracy of the other baselines in different metrics. And MegIS achieves all these benefits while incurring really small area overhead, which is only 1.7% of the area overhead, and also small power consumption of 4.6% of power consumption of the 3 ARM cores that are in a simple SSD controller. And there are, of course, all other units on the SSD controller as well. But even compared to these ARM cores, the area and power consumption is really low. So before I conclude, I would also like to talk about interesting result that shows actually potential of these storage-centric systems. And that is the cost efficiency. So let's see how it works. So we consider 2 systems. One is a cost-optimized system with a cost-optimized SSD and 64 gigabytes of DRAM. And the other solution, the other system is a performance-optimized system with performance-optimized SSD and 1 terabyte of DRAM. Okay? So we run these performance-optimized. We run the software baselines in the 2 systems, and then see how they will compare to MegIS running on the cheaper system, right? So here we see that when they're run on the same type of system, meaning all of them are run on the first, like the cost optimized system, MegIS provides life benefits. But also we see that it outperforms the baselines even when running on a much less costly system, right? So that's actually the point we would like to kind of like in this work emphasize with this result is that MegIS improves system cost efficiency, and we hope that it will make accurate metagenomic analysis more accessible for its wider adoption. Because it's the field in which we need to work with a lot of data. The amount of data we need to work with is increasing very fast over time. And to just continually rely on conventional systems and scaling them up does not provide an efficient way to widely adopt such useful, let's say, applications in larger scale, right? So there are many more results in the paper, like how would the performance look like if we rely on existing cores that are on the SSD controller, and how would all of this work if we had the same designs and optimizations but outside the SSD? So what's the significance of having this inside the SSD, right? Because we had a lot of hardware optimizations and software optimizations, and some things were also executed inside the SSD. So what's the exact benefit of all of these optimizations? So we clearly talk about it by showing like more, let's say, fine-grained results in the paper. There are a lot of sensitivity analysis, varying database sizes, memory capacity, number of SSDs, number of SSD channels, number of samples being analyzed at once. So, and for the other task, the abundance estimation, right? And that is the paper. Please check if you're interested. So to conclude, metagenomic analysis suffers from significant storage IO data movement overhead. And in this work, we propose MegIS, which is the first storage processing system for end-to-end metagenomic analysis that leverages and orchestrates processing both inside and outside SSD, or like in general, also storage system, and it improves performance, energy efficiency, while maintaining high accuracy and incurring only low cost. So yeah, that will be MegIS. And also I have been in the group with many other people working on different directions. Here is a summary of it. So if you're interested to get in touch, please let me know if you're interested in working in any of these directions. And yeah, thanks for listening.